हाँ, इस बुक पर Hello, everyone. Good evening. Can you hear me? I'm audible to all of you.
Yes, ma'am.
Okay, thank you. So let me welcome you all for the today's session, which is the first session on the industrial biotechnology, which will deal with the introduction part. And in this course, we will be having a total of 12 sessions, which will include the fermentations, enzymes, application of metabolic engineering, and certain case studies related to different sectors where industrial biotechnology has played an important role. So let us start today's session, that is on Introduction to Industrial Biotechnology. Let me introduce myself. I am Dr. Anshu Singh. I will be your course instructor for these 12 sessions. And during this course, I will be giving you some assignments also, so that you will be getting a clear picture of how you can have a better understanding of industrial biotechnology. So let us start with introduction part. We all know the human activities like industrializations, urbanization, such as petroleum, extraction of the minerals, has resulted in a lot of pressure on our environment. Okay, and this have a profound impact on the world environment as well as on the quality of life. And if we see this major part of this petroleum extraction industrializations, all are mainly chemical based. Okay, though chemical based industries are the backbone of our global economy. Okay, most of our day starts with our products which are chemical in nature. Okay, so we can't replace chemical suddenly. We have to go for the processes. We have to design the processes in such a way that we can minimize the effect of the chemicals. And thus, we have to save our planet, we have to save our people, and at the same time, we have to meet the economy so that the world will not face any critical situation. So the term comes sustainability. Sustainability is a very common terminology which is used in all the fields. Okay, if you talk about socio-economic, then also sustainability is there. If you talk about different sectors related to your, you can say pharma or everywhere, sustainability is now a prime importance. Why sustainability? How can you define a sustainability? Sustainability is action or the strategies which we make so that nobody understands. So I was talking about sustainability. Sustainability is actions, or you can say strategies, which we make to meet the need of the present, okay, without spoiling or without affecting or the compromising the needs of the future. Like simple example is that if you want a something, like if you want a simple, I will give you an example of Ganga, okay. Ganga earlier, if we look back in the past, Ganga water was pure, okay. With the time, with our demand, with the demand of human civilization or that, what we have done, we have deteriorated the Ganga water, okay. There are lots of heavy metals, there are lots of chemical waste which are going into the Ganga water. So what will happen in future? Does this future, in the future, the Ganga water can be used by your future generation? No, because it will be spoiled. So we have to take
I mean,
mic is on, kindly mute your mic.
Huh? Nam juniors.
So we have to focus in such a way, we have to design a process in such a way, industrial process in such a way where we can sustain. Kindly mute yourself. I'm requesting every time, please mute yourself. So we have to make processes which are sustainable, and during this whole process of industrial biotechnological course, my prime aim will be to make you learn the basics of sustainable processes for bio-based products. So we are targeting the bio-based products, and how these bio-based products can impact on a global sustainability. When we talk about the profit or economically viable, we are talking about creating a value and the wealth. When we are talking about the profit economy, we are talking about the value and wealth and profits. When we are talking about planet, that is saving our earth, we are talking about creating a cleaner, eco-efficient products and processes to prevent pollution and depletion of natural resources. When we are talking about people or a society, we are talking about to make to have ethnic behaviors in an ethical manner, and we have to provide jobs also for the people in society if we are creating or designing any process. So in all this sustainable process, we have to think about a industry which is sustainable when it produces goods and services in such a manner as to meet the needs and aspiration of the present. Means all the products and the processes if industry is want to be sustainable should meet the present, as well as it should not compromise the ability of the future generation to meet their own need. So how can a sustainability can be achieved? Okay, so if we are as a biotechnologist or as a microbiologist or as a basic biologist, what we focus on excluding the chemical process and including the biotechnological approach. But suddenly we can't replace the whole of a chemical process. If you look at the report by OECD in 2009, the bio-based process has increased, is slowly increasing, means it has from 2% in 2005, it can increase to 25% in 2025. So this is a slow increase. Suddenly we can't replace the whole of the chemical process. So we have to move in such a manner where we can modify the processes. We can include the zero waste concept, where nothing can be regarded as waste, because most problem related with chemical processing industry is the waste generation. So we have to design a biotechnological approach on the zero waste concept, where we will be using all the waste which are generated in the process as a feedstock or development of a new bioproduct. In 2015, if we look at the biotechnological approach, the 50% of all medicines were from the biotechnological approach. And in the way, if we are including biotechnological approach, the carbon emission will be spared by 33 million tons if we use industrial biotechnological approach in our industries. So let us define what is biotechnology. Biotechnology is a tool, okay, where we adapt or modify the biological organisms. These biological organisms can be yeast, fungi, bacteria, plants, and enzymes. And from there, we design a process, we design a system in which we develop a process in a very eco-efficient way, which is not only more profitable, but also more environment friendly. Okay, so this is a biotechnology where is a development of a bio-based products by where we adapt or modify our cells or its components for getting a desired output. And these biotechnological can be a fermentation technology, where we are using the microbes for getting a biotransform products, or we are using the microbes for getting our desired outputs. Then we have industrial catalyst, which we are called industrial biocatalyst, where we use enzymes for the bioconversion. Then we have a plant biotechnology, where we are using plant tissue culture techniques, or we are modifying the plant cells by RDT to get a desired products. And we have animal biotechnology also, where we are focusing on getting our bio-based products. Okay, most of the time the animal biotechnology products are related to vaccine, antibodies, and certain gene therapy is related to our animal biotechnology. So all these fermentation, enzyme technology, plant biotechnology, animal biotechnology, we have come across. Okay, we have come across these when you will be studying, or when you are still you will be studying, you will knowing that these are we do in our lab process also. Like if you will be going have your fermentation lab, you will be growing your bacteria, you will be growing your fungus. In the enzyme, you will be using certain enzymes for the conversion. But these are we do in a lab scale. Okay, we do in a small scale. But when we use these technique this adaptation or the modification or the steps to increase the bio-based products at industrial scale, that is called industrial biotechnology. And the color which has been given to the industrial biotechnology is called white biotechnology. Okay, so there are different color codes which are given to the different aspects of the biotechnology. But for the industrial biotechnology, the code given is white biotechnology. So how can we define white biotechnology? It is an application of efficient techniques at industrial scale. So the difference is what in the comparison to if we look at fermentation, enzyme, plant or animal, the same process which you are doing in the lab, now you will be doing at the industrial scale. Means you are focusing on the commercialization of your desired product. The main prime importance of your industrial biotechnology are you are focusing on the commercialization of your product. Okay, so now you you're producing that at industrial scale, and you are getting a desired end product by using same concept of biotechnology, that is viable microorganisms, cells and tissues of plants or animals, or even certain functional components of the organisms like enzymes. Sometimes DNA is also used for certain cases in fully controlled conditions. Fully controlled conditions means every process is fully optimized. Means you can regulate each of those processes. There is a pH can be regulated, the heat generation can be regulated, temperature can be regulated, and all this regulation is done in a fully automatic bioreactor. So the main aspect of industrial biotechnology is your bioreactors. Bioreactors are the main center point for your industrial biotechnology, where you will have to optimize or you have to control all the conditions in the bioreactor to get the desired end product yield or productivity. Okay. So what is the objective? Our objective when we go for any industrial application, our objective is fixed. We want to get any bio-based product or you can any bioproduct. These bioproducts can be of food related, that is a fermented product, single cell proteins, which is obtained from yeast or algae. Then there's citric acids, vitamins, amino acids, or pharmaceuticals like insulin, vaccine, drugs. These are our products. Or they can be a biofuels like biodiesel, bioethanol, or biobutanol. These bio-based products can be any of these. Objective will be any of these. If you want to go for insulin, your process will be totally different if you're going for a food fermented product. So first you have to fix your objective. What exactly your end desired output product? You have to fix your desired output product. So you will have to fix what you want. Now, once you have fixed your output, the next step will be, what will be the methodology? How will you proceed to get this bioproduct product? Okay. So when we have to choose, there will be number of methodologies, there will be number of processes, and we have to go for a greener approach. That is a sustainable process. Means we have to go for the front of the pipe approach, that is designing the process with a minimum waste and better yield and productivity. So if we want an industrial biotechnological sustainable process, we have to go for a minimum waste generation with a better yield and better productivity with a better profit. Okay. Sometimes it can happen that you want to get your product from a waste which has already been generated. So these waste can be from a chemical industry also, and you want to generate a bioproduct from this waste which has already been generated. So in that case, that methodology will be termed as an end of the pipe approach. Okay. So the method or process which you are choosing for getting this objective, like you want to get a bioproduct, it will depend upon the process you are choosing. And the most important step will be what the feedstock you are choosing and what type of technology you are using. If you are going for fermentation based bioproduct generation, or you want an enzymatic conversion based bioproduct generation, or you can use, you want to go for plant cells. So you have to choose a method or a process which will generate minimum waste, better yield productivity. Okay, so these will be your sustainable process approach. Now, if I define a bioproduct, okay, so what is a bioproduct? Bioproducts are the products made with some component of biological or renewable material, okay. So bioproducts, if I say these are the products which are made by some component of biological or renewable materials, and the renewable biomass may be from agricultural crop, trees, marine plants, microorganisms, and some animals. So I have given you some list of bioproducts, okay. They can be a biopolymers. These biopolymers include your bioplastics like polylactic acid, polyhydroxyalkanoic acid, biofoams, which are produced by starch plate or gluten based. Then you have biofibers, then biocomposites. And if we talk about the biochemicals includes the bulk chemical as well as the fine chemicals. When we talk about the bulk chemicals, these chemicals are not that purified chemicals, but the fine chemicals are the more purified one. They have a high titer value. So these biochemicals includes organic acids, alcohols, ethanol can be included, aromatics like toluene, paraxylene, biodyes, natural dyes, bioadhesive. If we talk about, there are bio-based adhesives, biomimetic adhesives. So there in a biomedicine, if we talk about, there is a biopharmaceuticals, antibiotics, vaccines. There are botanical medicines like quinine, Plexitexals, which are produced from the plant's biotechnology extracts. Biocosmetics are there where bioproducts are used. Biopesticides are there, like biochemical pesticides includes pheromones, pyrethrines, and microbial pesticides, certain beneficial bacteria and fungi act as a microbial pesticides. And there are certain pesticides which are produced from genetically modified organisms. But sometimes, if you look at these list of bioproducts, these list of bioproducts are produced directly from the renewable biomass. They are not undergoing any biotechnological modifications. Let me explain you. Do you know about biochar? Do any of you know about biochar? I'm audible to all of you. Can you hear me?
Yes, ma'am. Yes, ma'am.
Okay. Till now, it is clear what I am telling?
Yes, ma'am.
Yes,
Okay.
ma'am.
Okay. So I was talking about like bioproducts, as I have told you, bioproducts can be any product which will having a component of biological as well as a renewable material. Okay. So if you talk about biochar is a solid additive which is used in the soil and it can be filler in cement industry, and it is animal feed. Okay. So these biochar is produced when you take a biomass. So you take agricultural waste, you take a forest waste, you do the thermochemical conversion in an oxygen limited environment. So here you are using a chemical process for conversion of biomass into a biochar. Okay, you are using a thermochemical conversion. You are not using any biotechnological approach. You are using a biomass and directly converting into a biochar by using a thermochemical conversion. Okay, so here you're using a biomass, you're doing a thermoconversion and you're getting a biochar, which you use as a soil additive, fillers in cement industry and animal feed. So these bioproduct is an industrial bioproduct. It is not an industrial biotechnological product. Are you getting my point? This is a type of industrial bioproduct which is getting from a renewable sources by chemical conversion into a biochar. So it is a bioproduct. But when we talk about industrial biotechnological product, we talk about, we have a renewable source. Okay, in that case, we have a renewable source which we tell us as a bioresource. Okay, so renewable resource, we take a renewable resource, then we do a bioprocessing using a biotechnological approach, and then we get an industrial biotechnology bioproduct. Is it clear to you? In an industrial biotechnology, the main aspect is using the bioprocessing technique, biotechnological approach to get a bioproduct. And in the meantime, in the process, if a bio waste is generated, we used in a form of a recycling. So all this process is a type of zero waste concept. Nothing is waste in this process. Is it clear to you what is the difference between a normal bioproduct and an industrial biotechnology product? In the industrial bioproducts, there can be a chemical steps included, but when we talk about industrial biotechnological approach, we are focusing on application of biotechnological steps only. So let us talk about where these biotechnological applications has been used. In the cosmetic and perfumes, if we look at the personal care ingredients have market topped $15 billion in 2019, and in which bio-based resources were about 20% of that amount. Okay, so the acetone, which is used in the nail polishes, these were produced by fermenting plant sugars. So the sugars which were produced by fermentations were used for the production of acetone. Okay, so it is a bio-based product. Similarly, palmitic acids that gives your hair that glossy shine, the glossy shine, is one of the most common saturated fatty acid which has been produced by the microorganisms and the plants. So you can see these bio-based products has been used in the personal care ingredient sectors. If you talk about a simple examples of the food additives and the nutrient supplements, you can see the omega-3 fatty acid, which is a polyunsaturated fatty acid, which is very much required for the healthy body development. It is mostly extracted from the fish. Okay, so if you can see, this is a marine microalga, and the process we depend upon the extraction of the omega-3 fatty acids on the fish. But by using the biotechnological approach, we can get directly the omega-3 fatty acids from the algal oil. And the company Selena is giving this production of omega-3 fatty acids through algal, you can say algal production. Similarly, if we talk about the plastics and other biomaterials, you can get these plastics and other biomaterial bioproducts from cellulose derivatives, polyhydroxyalkanoates, PHA, polylactic acid, PLA, and star blends. So these lactic acid or hydroxyalkanoates, these are produced by microbes itself. Okay, so the bacteria have a property to form a biofilm, and through this biofilm, they are producing a biopolymers. And Ecolife, which is a firm that is based in the Chennai, are using this concept of bioplastic for industrial packaging. So there are mostly top 4 firms which are working in the area of bioplastics in India. And they are focusing on utilizing these concept of industrial biotechnology, getting a biopolymers from the microbes and using them for the packaging purposes. Okay, they have Ecolife have also used this bioplastics for lamination also. So you can see that how this industrial biotechnology is playing an important role. If we look at the bioenergy sector, you all know the bioenergy sector is a most promising sector nowadays. And the 2 important components of the bioenergy, you can say biofuels, are bioethanol and biodiesels. Okay, so there is a blending, you know, there is a government of India has proposed 20% blending of ethanol in petrol, and they have also reported 5% blending of biodiesel in diesel by 2030. So you can imagine that these ethanol and bioethanol and the biodiesel, which are produced by industrial biotechnological aspect, using the concept of fermenting the sugar and then using the you can say digesting the cellulose into the sugar form and then fermenting the sugar into ethanol form to get your bioethanol is an important industrial aspect. And you can see that there has been to 2021 only, the India has targeted to meet the 20% ethanol blending in petrol. And similarly, the biodiesel, which we get from our algae, because they have a property of storing the lipids. They are known as lipid producing organisms. So in a stress condition, they produce a lot of lipids, and these lipids and through esterifications can be converted into biodiesel, and through this biodiesel can be get, and this will be used for the 5% blending in with diesel by 2030. So you can see if Someone is targeting to set up a bioethanol industry. So they have to focus on how they can have a proper design of the processes to get a maximum yield in a cost efficient, environment efficient, and eco efficient manner. So bioproducts can be of three types, okay, depending upon their value and depending upon their volume, okay. So first, if we look at the high volume, low value products, the bioproducts which are produced in very high amount, okay, that is produced in millions of kg per year. These are the products which have a very high volume. And since they are produced at very high volume, so they have a very low price. So they are called as high volume, low value products. And these are mostly lactic acid, citric acids, or the chemicals which are required in the bulk, okay. And why they are, because they are high volume, because there will be high conversion rate, high yield, and high titer, means their purification is not much required. So in a high volume, low value product, you don't go for most of the downstream processing steps. You just go for a maximum yield, high rate of conversion, and high titer in the production step itself, okay. Next type of bioproducts are those which have a medium volume, medium value products. So they are required in very less amount in the comparison to high volume, and they are produced less than millions of kg per year, and they mostly includes antibiotics, single cell proteins, vitamins, where purification matters, okay. So there you have to include the purification steps. Next, if we talk about low volume, high value products, they are usually required in high amount, but their production is very less, okay. So in this case, the prime importance when we go for production, we majorly focus on stability and level of expression, and utility cost is not a major factor. So the major factor which people focus on when they are producing a high volume, low volume products is that the product should be stable. And as you know, the low volume, high value products include your vaccines, insulins, and antibiotics, okay. And if I give you a simple example of low volume, high value products, you will be all knowing about the recent certain parents having a child, they are suffering for spinal muscular dystrophy, which is a rare genetic disorder. And it's one injection cost rupees 16 crore, okay. So this injection is dolagenisima. And what this injection consists of? It consists of a healthy copy of the faculty gene, okay. So what this does, which allows nerve cell to then start producing the needed protein, which is required for the proper functioning of the muscles. And this drug has a 14-day shelf life. So you can imagine this product, one injection is costing around 16 crore, okay. So this is a low volume and very high value product. Is it clear to all of you? So is it clear to all of you that how bioproducts are divided on the basis of their volumes, their values, okay? Is it clear? I'm not getting response from any one of you.
Yes, ma'am.
Yes, ma'am.
Kindly respond in the chat box so that I can understand. If you have any query also, you can just ask me, okay? Till now, is it clear to every one of you how we can divide bioproducts in different sections related to their application and related to their, okay, thank you. So I was talking about this low volume, high value products. You can imagine most of the vaccines which we are in the market, they are having a high cost because they are produced in very low volume. Their purification cost is very high. And the most important thing, the process which they focus on the stability, the vaccines or the antibodies, so all should have a proper stability so that they can be used. So most of the cases, it has been found in the spinal muscular dystrophy that person has been given the injection, then also they were not cured. The problem was that the drug has a very small 14-day shelf life. Okay, so during the period of this transportation from US to India, and during the custom issues, your injection lost the shelf life. Okay, so it has been reports that the stability of these low volume, high value products is very important. Okay. So now you have your bioproducts, you have an understanding of bioproducts. Now we will be talking about how we will scaling up this bioproduct. Okay. So industrial biotechnology is all about scaling up. So scale up, if we define, scale up is a process increasing something in size, amount, or production. Okay. So when we are talking of increasing something in size, we are talking about the batch size. Okay. When we are talking about the amount, we are talking about the different yield. And when we are talking about the production, we are including both batch size and different yield. Okay. We are focusing on both. So let us understand what is a batch size. Batch size is that you have a specific quantity. You have focused that you desired this yield only, this quantity only of your product, and you have a single manufacturing order, and you have a uniform character and the quality within a specified limit. Means you have fixed everything, that there will be a finite period, there will be a starting point, there will be ending point. Okay, and in this part we will get a desired amount of yield. Okay, so what you have done, you increase just it. Like first you have taken a 10 liter, again you are giving a 10 liter, 10 liter, 10 liter, so that you get a desired amount of yield. That is called batch size increasing. Fixed amount of size you are increasing. Okay, so this is called batch size, where for a finite period with a definite starting and end point and time variable with the operating period, you get your yield. But when we want to focus on amount, when we are focusing on amount, we want to increase the yield. So we have to change the batch size. Okay, because we want a different yield, so we have to change the different batch sizes. We have to change the size of our production. And when we will be changing the want a different yield, we have to focus what we have done at our lab scale. Okay, so when we work at our lab scale, we have a fermenter. I'm talking about a fermentation technology now. So we have a fermenter of approximately 0.5 to 10 liter. Normally, when we work at the lab, what we do? We take a flask, or we take a certain polythene bags, or we take in a certain bioreactor, which is of size of 1 to 5 liter. We vary the our microbes concentration. We use the feedstock, we add the media, and then we go for keeping it in a certain incubation time. And we check at what incubation time, like at what day we are getting our desired output. Okay. And once we get our desired output, what we do, we go for extraction. And then after extraction, we go for the purification. But in reality, industrial scale bit differs. Okay. How it differs, that now your production unit is 10 to 100 liter. Okay. So you have done your process only for 10 liter. Okay. So in normal process, when you want to scale up, what you do, from the lab scale, you move to the pilot scale. Okay. So the pilot scale, the production unit is about 10 to 100 liter. Okay. So at the pilot scale is the most important aspects for the industrial biotechnology is the pilot scale, where the production unit is 10 to 100 liter. And after the pilot scale, the next steps comes for the demo scale, which is called demonstration plant unit. And it has a production unit of 100 to 10,000 liter. Okay. Once you have got a better analytical or better understanding of how your system or how your bioreactor is working at the demo scale, you will going for the full commercial scale with greater than 100,000 liter production unit. It is not that from the lab scale you will be jumping directly to your full commercial scale. You have to go by step-by-step process. Pilot scale plant can take about 0.5 years to 3 years for proper analysis. Similarly, demo scale will be taking another time period for getting your results. And the whole process from lab scale to full commercial scale takes around 10 to 20 years. Okay, so it's not that you have got a result at a lab scale, you will be giving to industry will be starting its production suddenly. No, they will be going for the pilot scale analysis, then they will be going for demo scale analysis, then they will be preparing a full commercial scale greater than more than 100,000 liter production unit. This is the process this is the step which is taken when your process is novel. Okay, sometimes if the process has been well established, that it has been used in various industries, so they just skip the demo scale. So from lab scale to pilot scale, they will be moving directly to your full commercial scale. Okay, so this is the step which we take during your in the case of the process which has been well established, which is not that novel, but in the case of novel process where it is first time you are using, in that case the person will be going for pilot scale, then demo scale, and then full commercial application scale production. So let us understand step by step. Okay, so lab scale is the discovery phase. Okay, where you have a R&D section, you have a platform to test and evaluate the productivity, and here you have a better handling and careful measurements, which is not is in the case of your pilot scale, or it is not in the case of your demo scale. So normally lab scale is called the bench scale, and it is the important early stage tool for assessing and scaling up of your technology. Okay, so once you have done your things at the lab scale, now why you should go for a pilot scale or a demo scale? There is sometimes it happens there are certain unexpected problems which occur in a project, and it will necessitate costly changes. So just imagine if you are producing, you are having a full commercial plant, there is certain problems are occurring, and at that time it is required to change the certain aspects of your bioreactor. So it will add more cost to your production unit, and which is not feasible for any industry. Okay, so that's why the industry focuses on ensuring that certain unexpected problems can be identified at pilot plant or can be identified at the demonstration plant, because they are small units. So here we can change the units or incorporate the changes which are required for the better yield. Okay, so pilot plant is the first window into continuous processing. In the lab scale, we are not focusing on the continuous processing. We are just producing, we are purification, we are telling that this is your yield. But pilot plant provide the first window into continuous processing, means your process is going on, your product is producing. Okay, so in the performance test are carried out to determine or confirm the yield and the data. Okay, so continuous processing will help you to understand that if this bioreactor is working at a full commercial scale, then what will be the problems which will be happening. Like sometimes what can happen that if you're working in the pilot plant, there is a heat generation. Okay, so if there a lot of heat generation will be there, enzymes will not work. Okay, in the process. Similarly, if there will be a lot of agitation, there can be a shear stress. Okay, so microbial cells can die. So in this, so you have to do a continuous process. You have to mimic the whole process of the full commercial plant at your pilot plant level. Okay. Normally, this pilot plant processes are done in the reactor size, as I have told you, 10 to 100 liters. And in the demonstration plant, This has a higher capital and operating cost than your plants, and typically employed when the process technology is already well developed. So once your process technology is fully optimized, fully regulated at the pilot plant only, then only you will be going to the demonstration plant. And most of the industry from the demonstration plant only, they started expecting their products, they're distributing their products in the market to understand the market survey. So before going for the full commercial plants, some industries, they make their products accessible to the consumers and accessible to the markets to understand the market demand. Okay, so I was talking about demonstration plants. So I was talking about that the demonstration plant, the product which are there, they can be used for your understanding the market survey, consumer demands. Okay, and once the demonstration plant unit, the industry have both the technological aspect as the marketable aspects, then it go for the full commercialization. Okay. So this was about the production unit. Okay, but when we take an industrial process, we will find that there are other scale-up process flow also. Like there will be lab protocol, you will be getting a plant flow chart. Okay, you'll be preparing a plant flow chart, and then you will be scaling up your process. So till now, what I was talking about the production unit. Like you will get a pilot plant, then you will be going for demonstration plant. Apart from that, there are certain units which are also required, and we have to link all this unit to get a proper industrial biotechnological working industry. Okay, so what this scale up each process flow includes, it includes the processing units, it includes the purification units, it includes the packaging unit, because when you are producing in a continuous process, when your product is continuously producing, you have to focus on its processing also, on its purification also, and its packaging also. Okay, so that there can be a better performability. Okay, so apart from that, you have to focus on your waste treatment unit. If it is solid waste, how you can be going to convert this, you can treat this solid waste, or you have any liquid waste, how can you are going for this liquid waste. Okay, and after this, you have a proper understanding of this, all these unit, processing unit, purification unit, packaging unit, water treatment unit with your production unit. You will link each process step. Okay, so each process step will be linked simultaneously, and then you will get a better performance. Okay, so scaling up is not only related to your increasing your production, but it also related to your packaging unit, your purification unit, your processing unit. Okay, so this is a way where your scaling up is done. Now, all of you will be knowing about cheese. Okay, all of you are knowing about cheese. Response, kindly just give me a reply. What is a cheese? Kindly reply in a chat box. What is a cheese? How is a cheese made? Any one of you? No idea. Is cheese is a biotechnological product or a normal bioproduct? I think you all will be loving cheese because it is used in pizza. So I hope that you will be having an idea. Yeah, cheese is a dairy product. Cheese is derived from a milk, right? But it is, yeah, cheese is a biotechnological product. Okay, cheese is a biotechnological product. And let me give you an overview how cheese is made. Okay, so this is a industrial production of cheese, how it is made. Okay, so you get a raw milk, and from the raw milk, what you do, you go for the pasteurization. You know, pasteurization is a technique where type of sterilization technique. Then after that pasteurization, then there is a standardizations. Standardizations means that since milk is gathered from a different area, so they will be having a different solid content, different protein content, different carbohydrate content. So before making a cheese, a proper standardization and filtration is done. Okay, and then after standardization and filtration, they are poured into a unit, you can say a bioreactor. And here there is an organism is added, that fermenting microorganisms are added, and after that, after the fermentation, the rennet enzyme is added. Okay, so that after the rennet, the addition of a rennet, there will be the formation of curd and whey. Okay, so there is a two biotechnological aspects here related. One is the use of fermentation technology, where they are using the fermenting microbes. Okay, so you can see the first step in the case of cheese production is use of the fermenting microbes. The another step is the use of enzymology. Okay, enzymes. That is a biocatalyst, industrial biocatalyst rennet enzyme is used. What is a rennet? Do you have any idea what is a rennet? Rennet is present in which, is extracted from where? This rennet enzyme, which is industrial enzyme. I'm not getting responses. None of you are responding. See, this is an interactive class. Okay, if I will keep on speaking, it will be not much understandable, because I need to understand, are you getting my points or not? Okay, no idea about rennet? Okay, let me tell you. So let us first explain this. Then I will show you a short video of Amul cheese making, and then we will be you'll be getting a better understanding of how cheese production is done. So this is a rennet enzyme. Then your fermenting microbes are added. Then once a curd is formed, you cut this curd. Okay, right, Anuradha, it is from animals. Okay, then after the cutting, they are stirred and cooked, and then this curd is the whey is removed, and this curd is passed to a milling process. So till now, if you look at this process, these was the production unit. This after the standardization and filtration, coagulation, curdling, cutting, stirring, and cooking, this all were part of the production unit. Now you are moving towards the processing unit. Okay, so now you are moving towards the processing unit. You have got a curd, you have removed the waste, that is your whey. So the whey is your waste, which you have removed it, you have drained it. Now when you got a curd, you have moved to the milling process. Now you're processing your curd. So what you are doing, you are adding salt to it. Then you are pouring into the molds before packaging. So depending upon the choices, it can be square cubes or it can be a different types of shapes. So you will be using a molds for this your cheese. You will press it, then you will keep it for ripening, and then after the packaging, you will be giving into the market. So you can get a fresh cheese like cottage and cream cheese, and after ripening also there are different type of cheese which are there. So you can see this is a simple example. They are scaling up where your processing unit is there. There is a waste drainage unit is there. Then you have a pressing and molding unit is there. So at every step you have to go for the analysis. And if you want to go for a pilot scale production of the cheese production, full commercial scale, first you have to run a pilot scale to understand that what step there is an unexpected problems can be generated. Okay. So as I have told you, the fermenting microorganisms have two things. That is, there is two steps of biotechnology, that is fermenting microbes and rennet enzymes. So let me tell you what are these two. So cheese types normally depends upon the culture we are adding. Okay, So for the hard cheese, thermophilic mixed cultures, Streptococcus salvarius, and certain lactobacillus are used in the ratio of 2:1:0.0 to get a hard cheese. Okay, when you want a soft cheese with fungus, you can use Penicillium candidum, Penicillium camemberti. If you want a soft cheese or semi-solid cheese, the fermenting microorganisms will be mesophilic acidification bacteria. So depending upon your cheese, your starter culture, that is the fermenting microorganisms will be different. When we are talking about rennet enzyme, okay, rennet is a complex set of enzymes which is produced in the stomach of a ruminant manus, means the stomach lining of a calf, eat, or baby goat. So these enzymes are extracted from the stomach lining of a calf, kid, or baby goat. Okay, so this is a complex set of enzymes. And how this rennet do? Rennet is the second phase, which is a coagulating phase. So as it is an enzyme, so enzyme will be have optimal temperature and the pH and the salt bridges. So what does rennet do? Casein, you know, casein is there, the protein. The rennet changes this into the para-casein, and in the presence of calcium, they form a gel-like structures. Okay, and so these gel-like structures are the form of the curds which are used in the process of producing a cheese. So rennet, first phase is your first phase is the rennet is microcase by para-casein, and from the para-casein, the gel formation takes place in the presence of calcium salt. Now, the another important thing is that most of the time people don't prefer the source like rennet as it is extracted from animal source. So there are certain plant-based rennet has also been derived, and certain microorganism-based rennets are also there, where rennet is produced from the microbes. So let me show you one. Let me show you one production unit of a cheese to make you better understanding. For a better understanding, just wait.